First, I would like to thank the organizers for inviting me to give this lecture. Let me share my slides. I'm going to talk today about molecular understanding of electromagnetic field biometer interaction for rational design of biochemical sensing devices. My name is Michal Cifra. I am from the Institute of Photonics and Electronics of the Czech Academy of Sciences. Let me start with giving you a vision we follow. So we are asking this question: how far are we from a real medical tricorder? You might be familiar with the science fiction series Star Trek, where there is this funny device which can send some waves and also receive some waves from the organism. And in this way, it can diagnose what's happening inside the body and also treat particular diseases. So this is actually kind of science fiction vision which drives our scientific research. In our research team, we are exploring how external electromagnetic field interacts with organisms and how we can engineer this interaction. And also we are exploring how organisms generate electromagnetic field. So putting these to the words, our mission is to probe and influence biomolecular systems using an electromagnetic field. The vision which we currently follow, given by the Star Trek example, is to bring foundations for novel electromagnetic methods for gentle and more efficient bio-nanotechnology and medicine. The current tools which we are using are advanced electromagnetic concepts, which intensify electromagnetic field biometer interaction. Then we also use micro-nanotechnology enabled tools and computational methods. So we typically start with the theory, which enables us to understand these passive and active electromagnetic properties. That means how external field is acting upon the matter and also how biological matter generates electromagnetic field. Based on these predictions from these theories, we generate hypotheses then, which tells us at which frequency ranges and power densities we have to use to make the experiments in. In order to verify these theories, we develop particular technologies, often based on the micro nano fabrication of the chips, and also we are building whole experimental platforms, all this backed up by the electromagnetic design. And then we perform experiments which verify the theories and hypotheses. And mostly we are working on the level of the subcellular protein structures and the cells. Our research team, Bioelectrodynamics, is our name, and we are a bunch of the people from different backgrounds, ranging from engineering through the biochemistry and up to the biophysics. So let me give you our perspective of how we can rationalize the radio frequency and microwave chemical and biological sensing. So the principle which we consider is the following. So let's imagine we want to sense something in the biological system. That could be a change or a difference of the sense parameter compared to control or developing over time. So that typically reflects into the change of permittivity, complex permittivity, the electric properties of the whole system which we measure. And then it's typically reflected in a change of S parameters. Well, which we can also simplify just measuring the resonant frequency in case of resonant sensors, or we can have a look on different parameters. But in general, it's the S parameters which we measure in the complex measurement. What I would like to focus on today in this talk is how we can understand what's happening in biological samples from a molecular perspective, and how does this bring about the absolute values and also relative changes in dielectric spectra or complex permittivity spectra of these systems to be probed. However, these are frequency dependent parameters, observables, this permittivity or change of the permittivity due to some process we want to sense. And therefore, the central question which we ask and try to address today as well, which frequency or frequency range is optimal for sensing of particular parameter to be sensed, and by this we mean where we expect the maximum change of the permittivity, either real part or imaginary part of it, of the system, due to the processes we want to sense. So let's give first a very brief introduction into the dielectric properties of biomaterials. So we know that um, water-based materials, that's what organisms are mostly, are their permittivity spectra are governed by rotational diffusion processes, which also determine the time scales of the spectra. And these are typically described by a dBi or other derived models. And for example, in very simple case, just sort of the pure water, we see that these are the important parameters. It's the static dielectric constant of the water, or the dielectric increment, and also it's the time scale rotational diffusion, which then determines the frequency of the peak of the imaginary constant of the imaginary part of the permittivity. The full line is the real part. The dashed line is the imaginary part of the permittivity. Now, when the system gets more complex, for example, we are adding some biological components, their biomolecules to the water, the permittivity spectra change. For example, when we are adding a small polar biomolecules, like we are going to give examples in the future slides, what we see, there are another parameters, let's say another terms appearing in a typical dBi model, and that adds additional increment of the real part of the permittivity at the low frequencies, and also there is an additional peak arising in the imaginary part of the permittivity due to the biomolecular properties. Now, how we can predict, at least semi-analytically or half empirically, these basic parameters of these models? That means these increment static permittivity and the parameter tau, the time scale of which determines the peaks of the imaginary part of the permittivity. Getting back to the textbooks, basically, the dielectric increment is determined by the density of molecules and by their dipole moment these molecules carry. So these are the most important parameters which determine how much particular molecule will contribute to the real part of the permittivity. And the time scales related to the rotational diffusion of these molecules then determine the peak, the frequency of the peak in the imaginary part of the permittivity. And this is determined by the volume, effective volume of the molecule, and effective solvent dynamic viscosity. So these are the simplest analytical equations, considering some assumptions as a spherical molecule and so on. Well, the problem is that the analytical equations for these parameters, they start to fail for a complex shape of biomolecules, for multi-conformational biomolecules, the multi-component media, and if you increase the concentration of biomolecules and they start to interact, these equations start to fail and they do not give very accurate predictions. So how we can do better than just using, well, rather old analytical equations when the systems are too complex? There is an approach which is now highly used in different research fields, and that's so-called molecular dynamic simulations, which employs classical physics. Basically, in this approach, we are solving the time-dependent equations of Newton equations of motion for every atom. And well, this is, as I mentioned, widely used in biophysics and also theoretical chemistry. And particularly for our purpose, this approach enables us to understand how macroscopic dielectric properties arise from molecular structure and dynamics, and also how electromagnetic field can affect biological systems at the molecular level. And there are many free tools available for running these simulations. So to give you an example, how can we use this to predict and understand the permittivity? So let me give you this example. In our case, we just simulated a very simple system of water molecules, these small red and white flickering V's. And then we also had the system, the biomolecules, the alanine molecules, if this effective concentration. And the blue flickering arrow is the dipole moment of one of the components. Either it can be dipole, what we can follow is dipole moment of the biomolecules, or dipole moment of the water phase, or the correlation of these two. So now when we increase the concentration of these biomolecules in the water, the time scale of this simulation is roughly a few nanoseconds. Then, based on the trajectories which we obtain from these simulations, we can calculate dipole moment autocorrelation function. autocorrelation function of dipole moments of these individual phases. And when we have this autocorrelation function or the cross correlation between the components, then we can use this to calculate how these individual components contribute to the susceptibility, that which is directly related to the permittivity. Now, what are the important findings from these simulations? So you can see how these outcomes of this simulation basically look like. So these enable us to predict purely computationally based on the atomic structure what will be expected permittivity. The absolute values are not perfectly matched, but we have very interesting also qualitative insights. One of the insights is that increasing concentration of the biomolecules, here alanine, what happens is that the imaginary part of the permittivity, the peak of it, is decreased to lower frequencies. What is another important finding, which was most striking to me when I saw the data some years ago, very first time, that when we increase concentration of biomolecules in the water-based solution, what starts to be increasingly important for the dielectric spectra is not only the contributions of individual components, here the alanine, amino acid, and the water, but it's also the cross-correlation contribution between the motion and dynamics of these two, which makes a significant contribution. Here is it in a dotted line. So now, having these basics, I would like to show you a few examples how we can rationalize the design of sensors. So at first, I would like to give you one example, how we just a design example, how we could potentially increase the sensitivity, for example, for probing hydrogen bonding and water molecule dynamics in a biomolecular systems using microwave sensors. So from the findings I showed you earlier from these molecular simulations, we know that when biomolecular concentration increases, it usually changes, it increases the relaxation time of any molecule present in the system, which then causes the frequency of the peak of the imaginary part of the permittivity to shift to lower frequencies. This means increasing the rotational diffusion time, which determines the frequency of the peak. Well, then if we want to sense this change, I have to ask, so at which frequency will be the largest change actually taking place? So basically we subtract the curves and we take absolute value of those. And we found out that the maximum contrast in the imaginary part of permittivity will be somewhere around 35 to 40 gigahertz at a high frequency range. We selected this one for a particular reasons. We could also go for low frequencies though. Now, what does it tells us for the design? So basically here on the property of the biosample, in this case, simply the dynamics of the water molecule present in the biosample, this tells us at which frequency we can expect the largest change of the permittivity, and that should be the ideal frequency where we should target our design. So basically, this is basically told by this equation. We should focus on this frequency band. That's the frequency where the change of the imaginary part of the permittivity is the largest. In short, we designed a particular coplanar waveguide based periodic structure where basically we formed a so-called electromagnetic band gap, which behaves as a band stop for a particular region, and there we introduce a single defect which causes a sharp resonance, as you can see in the transmission on the reflections spectra of such a structure. So this is how the structure looks like. Well, first we did analytical predictions, and then we made full wave simulations where we optimized the structure. So you can see that the frequency where the waves cannot propagate because there is a kind of band gap or band stop, the waves from the right basically are not being propagated and they do not go through the structure. However, at the frequency of where the defect is actually causing this resonance, at this frequency, the waves can propagate through the structure, and because of the band gap and also narrowed gaps here, there is increased interaction, there is increased electric field just in the middle where we inserted the defect. So this is how the structure looked like schematically. And what we basically showed when we loaded the structure with the microfluidic channel in the simulation is that when we run water or a sample through this channel and we change in the dielectric model in the simulator the relaxation time, then we can see that the quality factor of this peak is changing proportionally to the dynamics of the water molecule, the time scales of the water molecule. So in short, this approach based on molecular dynamics simulations enables us to predict where we can expect the largest permittivity changes. And there we design, well, we could use different designs too, different resonant designs, but here we selected to use, we decided to use so-called defected band gap, electromagnetic band gap device, which enables us to generate a strong resonance and the field compaction and the particle size side of the device. And there we place the sample, and we found out that we can correlate the dynamics of the water molecules with the quality factor of the resonance, which then actually enables to sense these dynamics of the water molecules. Another example is that what we could demonstrate it, how we can use these approaches to optimize the detection of the amino acids and their dynamics. So in this case, we focus on a dielectric analysis of the solution of the alanine. Alanine, which I already mentioned, is one of the small, very common small molecules. It's the most common building block of proteins, which are molecular machines responsible for life in a sense. And going now to the molecular structure of this molecule, because it's a priori known, then we can predict what will be the effective time scale of the rotation of rotational diffusion of this molecule, which carries the dipole moment, and that will then determine with which frequency we can expect the largest changes when this molecule will be added to the solution. So basically, based on knowledge of the size, basically volume, and the effective solvent viscosity, we found out that the target central frequency should be 4 gigahertz. However, we also want to have some sensitivity for the broader spectrum, so we had to make some optimization because of that. But basically, these are the basic numbers to follow for an 18 gigahertz. At this case, we designed the sensing structure based on the conductor-backed coplanar waveguide, where we made first analytical estimates of sensitivity by formulating the transmission model by analytical equations. So we can see that there's the coplanar waveguide's cross section, and this is where we placed our specimen or sample. Now we ask the question how we can optimize the geometry of this sample in order to probe the sample at the optimal frequency, which we determined earlier, this 4 gigahertz roughly. So we found out that the effective length of the sample has to be around 6 millimeters in order to have high sensitivity at 4 gigahertz while having reasonable sensitivity still at 18 gigahertz. Here you can see this is the reflection and the transmission based sensitivity. So based on these approaches, we could determine what will be the optimal length of the sample in order to maximize the sensitivity at particle frequency. So this is how the design before fabrication looked like. So we have this inverted coplanar waveguide design where we launch the waves on one side, then we let the wave traverse through the wires to the sample and then traverse back. So this is how the sample looked like. Basically, we get rid of the need for microfluidics in this case because we have this geometry is insensitive to accurate shape of the sample. So we don't have to care about particular shape of the sample as long as the field is confined within our sample. So what we found that using our chip, our sensor, we can use 20-fold lower volume than the reference bulk method to obtain the permittivity. You can see how the results look like for a pure water as a calibration standard, and you can see we have a very good match of measurements from our chip and from the reference bulk measurement. And also you can see that there is quite a reasonable semi-quantitative fit of the molecular dynamic simulations for this case. You can see then also the errors here. Furthermore, then we added alanine and we increased concentration of it. And we found that our chip performs reasonably well. And we have very good detection of the permittivity of these solutions. And we also reasonable match to molecular dynamic simulations. So take-home message for this part is that we developed and tested a chip and method to obtain complex permittivity of liquid samples within this frequency range with 20 times lower volume than commercial methods. And here we first use analytical estimates to see where we should have the highest sensitivity for sensing the presence of alanine and its dynamics, but also then we use a posteriori molecular dynamics to closer interpret the complex permittivity data to match it to experiment. Last example I would like to show very briefly is how we can use microwaves to sense some more biologically relevant structures, and these are so-called microtubules. So what are microtubules? These are very interesting biological polymers present basically in every cell, and these fibers, or actually they are microstructured fibers, basically they are tubes. They're essential for life processes because they enable cells to divide and also transport cargo inside the cells. And the particular feature of microtubule is they dynamically self-assemble and disassemble, or in chemical terms, they polymerize and depolymerize. So this is a very important process, which also determines how they can respond to drugs and so on. And therefore, one of the important parameters for this structure is to be able to sense if the structure is polymerized or not. So basically, if it's still are the building blocks are there, or these building blocks are assembled to a full microtubule. So we are basically will be comparing the tubulin to microtubule, if we can sense that. So we first thought, does it even make sense to try microwaves to sense these polymerization states? So at first we thought, okay, so it's known from the literature that the polymerization in general of different polymers, not only biological ones, affects solution viscosity. This is known since many tens of the years and measured by viscometers. So that means if viscosity is changed, that could lead to change of a rotational diffusion of any molecule in such a system. So we decided to again use up the water molecule, and if then we thought about, okay, so if the frequency will be changing of this rotational diffusion will be changing, so we should probably have a look on this frequency, because that will be this is also close to timescale of the water molecule rotational diffusion. Then anyway, because there is a little known about the tubulin and microtubule dielectric constant in the liquid state, we decided that we have to make a broadband operation of this device. But basically the idea is that we knew that we could expect some changes because polymerization affects viscosity. So then this is the idea of the experiment. So we compare either tubulin or microtubules, which we deliver to our chip, which I will briefly describe, to our experimental system, and we then expect and would like to see if there is a difference in S parameters measured through this system. So we designed for this particular purpose a coplanar waveguide-based chip, which with these dimensions on the glass slide with the particular calibration kit, and with the tapered section where the microfluidics channel is. Then we fabricated these with the thin film lithography and with lift-off process. This is how the experimental system looks like. We have a mechanical probe station where we connect the chip with the high frequency probes, and we have the outlet of the microfluidics. So you can see the output from the vector network analyzer, and here there's the microscopy image of the channel of the chip. So what we have found is that although there is a numerically rather small difference in a transmission through the chip, where in the microfluidic channel there is a tubulin or microtubules. However, when we subtract these lines, that means we subtracting the transmission parameters from the microtubules, we can see a clear difference well beyond the error of the measurement. So this tells us that we can distinguish whether the tubulin is polymerized to microtubules or not in this microwave microfluidic system. Additionally, we also determined for the first time the permittivity of the tubulin microtubules in solution in this frequency range, and we could find then a posteriori where are the largest changes in permittivity, both in the real part and imaginary part for the microtubules compared to the tubulin and also to the control buffer and other parameters. So in short, we in this part we could show that the microtubule state was predicted to affect complex permittivity based on the rationale which included the change of the viscosity. And then based on these predictions, we knew roughly at which frequency range we should design the sensing structure, again, based on a coplanar waveguide. And then we used it to sense the self-assembly state or polymerization state of these important biological polymer, and also we first time delivered the broadband dielectric data for the microtubule systems. So to conclude, we believe that the insights into microscopic dynamics of biomolecules help to select the frequency range where the largest dielectric contrast can be expected, and also they enable to interpret the dielectric data. And we believe the future outlook for this research is to use molecular dynamic simulations which can treat also more complex biomolecular biological systems to optimize and personalize the performance of the biological and chemical microwave sensors. So with this, I would like to thank you for your attention, and I will be glad to answer any questions either directly or here through the channels you can see on the slide. So thank you very much.